the rather provocative title, Time to Recognise that Mitochondria are Bacteria.
All right, well, thank you very much for inviting me. This is probably the shortest notice I've ever given a talk at the SGM or any conference. Sort of yesterday afternoon, he asked me to talk about something to take the place of Siv Andersson. And after a few moments thought, I thought, well, I suppose I could give a talk. But what we have to recognise is that I actually don't have a track record in bacterial taxonomy. I'm not Siv Andersson. I don't look like this. I look like this. I'm a different sort of person. I will be talking a lot about Siv Andersson's work, but probably not drawing conclusions from it that she may not endorse. Actually, I said to Ross that I will actually act, if you like, as a court jester for the session and be provocative. And if we take it in that spirit and you recognise that I'm doing this at short notice, then I think we can make this work. So what I've done is I've rehashed a paper which I wrote a year or so ago, which was published in Trends in Microbiology, which you can download and read if you wish to afterwards. It covers pretty much the same ground as I'm going to cover today. So what I'm going to cover is taking us from this view that we are comfortable with the idea that mitochondria are organelles that are descendants of bacteria to say actually they are bacteria, and look at all the ramifications of that, and particularly look at some applications of that in changing our appreciation of the world and actually our understanding of important issues. So of course the mitochondrion has these bacterial roots. Before we talk about those, it's worth just stressing that mitochondrion really lies at the heart of eukaryotic cell biology. And eukaryotic cell biologists spend a lot of time looking at mitochondria, key roles in energy production, apoptosis, free radical biology, intermediary metabolism, and all sorts of other things. In fact, my whole interest in this arose because I used to share a train journey with a guy who worked on mitochondria, and I said, well, you're a bacteriologist. And he said, no, I'm a cell biologist. And I said, no, you're a bacteriologist. And I said, I'm a greedy bacteriologist, you're one of us. And so we went. But there's no doubt that this mitochondrion is fully integrated into the life of the eukaryotic cell and functions as a subcellular organelle. Now, the idea that there was a link between mitochondria and bacteria actually goes back to the 1890s. German cell biologist there, Richard Altmann, proposed that, he called them bioplaston. He proposed that these mitochondria were autonomous elemental life forms similar to bacteria. And during the early 20th century, there were similar suggestions from various other scientists. But it was in the 1960s, really, that the proposal that it wasn't the result of endosymbiosis and this so-called serial symbiotic theory came to prominence, largely due to the efforts, but not entirely of Lynn Margulis. Now, some of the ideas that she had, I think, were now accepted as a bit cranky or not acceptable anymore. You know, the idea that spirochetes became cilia and flagella. But the idea that bacteria were the originators of the mitochondrion actually stands firm. And the evidence for that and the acceptance of that over the last half century has grown steadily. And it's now recognized that there are extensive and deep similarities between mitochondria and their non-mitochondrial bacterial relatives. This is a table. It's a rather busy table, and I'm not going to go through it, but this is a table from the paper, just looking at some of the ways in which there are these profound similarities between bacteria and mitochondria. So a colleague of mine, Ian Henderson, that works on the BAM apparatus. Well, mitochondria have a BAM apparatus as well in their outer membrane. We have similar kinds of translocases in the inner membrane, insertases, and so on and so forth. I'm not going to go through all this. The key point that was one of the things that really was the tipping point in recognition was the recognition that mitochondria generally have their own genomes, this circular double-stranded chromosome, similar to most of what you most often see in bacteria. Okay, so we're all kind of comfortable now with the idea that mitochondria are descendants of endosymbiotic bacteria, but what if we actually throw caution to the wind and start saying that actually mitochondria are bacteria? And let's see where this reformulation leads us. The so-called Sapir-Whorf hypothesis says that the way we describe the world influences the way we think about it. So let's start describing it as it is and say mitochondria are bacteria. And so I'm inviting you to come and play with me for the next few minutes as we take a kind of thought experiment, if you like, and look at the ramifications of reconfiguring things that way. And there's a long history in science of just doing thought experiments, playing with ideas and seeing where they lead. So why should we actually say this? Why should we say mitochondria are bacteria and that's that? Well, Darwin, actually in The Origin of Species, made the point that our classifications will come to be, as far as they can be so made, genealogies. And that aspiration actually really came to fruition in the middle of the 20th century with drawing on the work of Willy Hennig and the idea of phylogenetic or cladistic classification. And this swept through the classification study of animals and plants, both living animals and plants and fossil animals and plants, and now has become the accepted way of doing things. And there are some key tenets in there which came in with this idea of cladistics. One of the most fundamental one is that classification should represent only the pattern of phylogenetic branching within groups of lineages. It shouldn't concern itself with patterns of evolutionary innovation, simplification, sometimes put down as the clade versus grade dichotomy. And the first cladists actually swallowed a Greek dictionary and came up with lots and lots of new terms that have permeated the field. The idea of monophyletic groups. These are the only valid taxonomic groups because they contain all and only the descendants of a single common ancestor. And what were known as paraphyletic groups are now deprecated because they only contain some of the descendants of a common ancestor, not all. Here's a figure from my book, Rough Guide to Evolution, which kind of illustrates the point here. So we have things like reptiles, something you learn in school. Oh, reptiles, they're a class, aren't they? Well, they're no longer accepted in taxonomic terms because they're a paraphyletic group, because you can't define reptiles on the basis of descent from a common ancestor without also involving birds. In fact, fish also gone by the wayside. We can't have a taxon of fish because that would include birds and mammals and so on. Of course, we now accept that birds are dinosaurs. And when the old style use of the term dinosaur in taxonomic terms really means non-avian dinosaur, because there are dinosaurs still out there. Chicken is the most abundant dinosaur in the world. We have to get used to that. There is an uneasy relationship between the traditional Linnaean taxonomy, with this idea of one size fits all, this hierarchy, phylum, class, order, family, genus, species, and this more flexible succession of clades that's defined by phylogenetic taxonomy. And there are some clades that don't really fit into the Linnaean scheme, like the amniota, for example. There are some people out there who actually are saying, let's just get rid of the Linnaean taxonomy entirely and replace it by a strictly phylogenetic phylocode. And there are battles going on out there among people who classify plants and animals over this kind of issue. So it brings radical rethinks. I mean, these kind of approaches, obviously, as the previous speaker used these terms, monophyletic and so on, these approaches have permeated into microbiology as well. And have been caused to abandon some of the terms that we are kind of familiar with, like prokaryotes, it's a paraphyletic group, deprecated, get rid of it. Mycoplasmas, the molecutes, well, they're now actually belong inside a phylum Firmicutes. So you've got soft-walled things inside the firm-walled things, and we now accept the mycoplasmas are kind of gram-positive, even though they actually lack a cell wall, based on their phylogeny. So what about mitochondrial phylogenetics? Well, there's a whole literature out there on this, looking at molecular phylogenetic approaches, and Siv Andersson here has been one of the pioneers. And all of these support a bacterial origin of mitochondria, and more specifically, they support an alpha proteobacterial affiliation for mitochondria. So here's a figure from a recent paper which shows the mitochondria here actually clustering within this group of alpha proteobacteria with the rickettsia and anaplasma. I can't even say it, but anyway, these other as sister groups within that phylum. And just to make the point, Siv Andersson, one of the pioneers here with one of her pioneering papers back in Nature 10, 12 years ago, actually pointing out that rickettsia was the closest living relative of mitochondria among genome sequenced organisms. Free living organisms, well, not free, non-mitochondrial organisms. And she's actually still working in this field. This is a paper I just turned up this morning as I was just checking up on what she'd been doing. She's actually been looking at the relationships of the mitochondrial clade to its closest relatives and found new things in the oceans. All of these studies suggest that mitochondria form a monophyletic taxon. Okay, there's a wide range of genome sizes, and there are some oddities within that clade, some structurally similar, functionally distinctive organelles, things called mitosomes and hydrogenosomes, but they're all in the same clade. And by the logic of cladistic classification, this means that mitochondria belong in the domain bacteria, or in common parlance, it means they are bacteria. So why are we quibbling? Why do we say anything different? Similar arguments, of course, apply to chloroplasts and other plastids. And if we're going to do this, and we should, we have to then change the way we speak, and we talk about bacteria. When we talk about bacteria, we should be talking about non-organella bacteria, perhaps in the future, based in the same, in the way that we talk about non-avian dinosaurs. Of course, we have to give a name. If we're going to stick with Linnaeus, we're going to have to give a name to this clade. What is its rank, though? That really determines what name, and there is some debate still as to what rank we should be giving this. If we call it, if it fits alongside the Rickettsiales, it should be called the Mitochondriales. If it's, wherever it is, it's going to have its own family, the Mitochondriaceae. These are neologisms I've made up. I think that they are provocative but valid terms that we might want to start using. Now, if we're going to follow this, some of you might start to be getting uneasy now. This guy is a raving nutter. But let's take this even further. So if we're going to say that these are proper bacteria, we want to, how many species are we going to say there are? Well, in our thought experiment, let's take the human mitochondrial reference sequence, and if we compare that with, say, the equivalent from the Neanderthals, we get 98% identity in the small subunit RNA gene. That would, on the 97% cutoff, that would say, oh, well, they're the same species. I know that some people say 99 now. 92% if we look at chimpanzee. So, it looks like there probably is one species of mitochondrion per one species of animal in this very limited survey. What are we going to call the human mitochondrion then? Well, let's just be creative and call it mitochondrion laxi after the most abundant source of human mitochondria on the planet, Henrietta Lacks, about more which later. But, this is just the beginning. Previous speaker spoke about wasting time on things. If you want to waste time, we've got a large job to do to classify all those mitochondria and give them species names out there in the world. Now, some of you are spluttering with indignation. Of course there are objections and counter-arguments to all this. Of course mitochondria aren't bacteria. They're organelles. They've evolved into something so distinctive and so fundamentally different that it makes no sense whatsoever to continue to classify them as bacteria. And, Willy Hennig be damned. This is just common sense. Now, of course, there's a counter-argument that comes from antiquity, the so-called Shipper-Theseus argument, which is actually there's a continuum that links what you would accept as bacteria to mitochondria. In the Shipper-Theseus, there was this ship where each of the planks was replaced one at a time until none of the original planks remained. But all the way through, there was a ship that looked the same. Was it the same ship? When did it change from being one ship to another ship, if it's not the same ship? I'll give you a modern example, just to bring the argument right down to rather basic things. I don't know if anyone recognises this group here. This is the Sugar Babes. And the Sugar Babes have actually changed their lineup several times. And so the original lineup of the Sugar Babes, none of those people are now in the group known as the Sugar Babes. Is it still the Sugar Babes? So what about, if we want to draw a line and say, right, there's a place where the mitochondrion stopped being a bacterium, where would we draw that? There are a number of changes during that transition, but are any of them distinctive or profound enough? Well, one thing they've done is obviously thrown away a lot of genes, genomic downsizing, in terms of gene size, genome size and gene number. And they've outsourced genes to the nuclear genome. But those same processes are going on in endosymbionts today. And that process of downsizing, and Siv Andersson has done lots of work on that, is very common out there. And there is, in fact, some overlap in terms of genome size between the largest mitochondrial genomes and average-sized bacterial genomes. You know, we still accept endosymbionts as bacteria, and we give them Latin binomials. So, you know, I don't see that this argument really has cogency. This genomic outsourcing, we see many mitochondrial genes migrating into the nuclear genome, and it's still ongoing. These so-called numpties, where the human mitochondrial genome gets pasted into the nuclear genome and decays away. But we now know that that happens with other endosymbionts as well, and that I just read a paper of Wolbachia genomes are continually being pasted into their host genome as well. And it's been questioned as to whether Carcinella rudi, which is now one of the smallest genomes, whether that is actually viable. Because if you look at the gene complement encoded in its genome, it can't actually make all the things it needs to make. And so people have said, well, actually, it must be importing something from the host. There's no firm evidence for that. But it's the same kind of argument that happens with the mitochondria. The mitochondrions outsource this stuff. But we've given Carcinella a Latin binomial. What about, I think Eugene Koonin was one of the first to suggest the idea that it's actually protein import is the crucial point. Bacteria don't import proteins from outside, whereas mitochondria import loads of proteins from outside. In fact, those import pathways appear to have evolved gradually. There are multiple protein sorting pathways, and there's some recent studies that chipped away at this apparent irreducible complexity. It wasn't like one day there was a bacterium that didn't import, the next day there was one that did import. It was a slow process. And in fact, there's been a study recently that shows that some bacteria, intracellular bacteria, incorporate proteins into their outer membrane that are encoded by mitochondria in the same cell. So again, are we going to throw away the classification of those and say, well, they're not bacteria anymore because they're importing a protein? That argument loses. I mean, one of the problems, of course, is that we're actually creating a TARDIS-like taxonomy, and this is where it gets quite mind-bending, because if we say that every mitochondrion on the planet has to be classified into a new species, a mitochondrial species, and all those lineages associated with millions of different eukaryotic species, everything from blue whales down to amoebae, all of that has to be reclassified. We're going to end up with so many species in that alpha proteobacterium phylum, it's just going to be mind-blowing. It stretches Linnaean taxonomy to a breaking point. I know the phylocode people say it's already broken, but you know, it is. You can argue that's a serious problem. The other issue is that it means that each eukaryotic species is going to have to be split into two. So this is Homo sapiens here, but hang on, there's a nuclear genome and a mitochondrial genome. We have to give them separate names, do we? Well, in fact, it's not that bad a problem because it's already been solved for lichens where the whole symbiosis is just named after the majority fungal component. So we could keep Homo sapiens as a valid description of the whole assemblage of nuclear and mitochondrial symbioses. Another objection is that actually there's some evidence of polyphyletic origins of the mitochondrial proteome. So although the proteins encoded by the mitochondrial chromosome are alpha proteobacterial in origin, if you look at all the proteins that get into the mitochondria that are imported, in fact, only a fraction of those, less than 20% of those, actually are alpha proteobacterial. Most of them seem to have been recruited from the nuclear genome, some from bacteriophage. So if you've got an organelle carrying so much baggage from outside of its original lineage, does it make sense to call it a bacterium still? Well, there are some comparisons with languages here. I mean, we still call English a Germanic language. If you got the Oxford English Dictionary and you looked at the etymology of all the words in it, you'd find that the majority of them would come from French or Latin and maybe some Greek. But that doesn't mean that English is no longer considered a Germanic language. Yiddish, there's arguments that Yiddish was originally a Slavic language that got re-lexified and took on this Germanic vocabulary. So if we're comfortable with the idea that English is Germanic, so what's the problem here? Final objection is on the grounds of utility, that actually, we don't need phylogenetic purity, let's just stay with what's pragmatic and works. And microbiologists seem to be very happy with the idea that we call Shigella Shigella. Even though it's not a genus, we know it's not a genus. In fact, it's just a pathovirus, an ecotype within one species, E. coli. So, what's wrong with a bit of taxonomic messiness? Now, I hope I've carried you so far with my arguments, but you might still be saying, why is this important? It doesn't really matter, does it? I think there are ways in which it does matter, and let's look at two of them now before I finish. Well, before I look at two case studies. Before that, though, just a bit more whimsy. If we accept mitochondria as bacteria, we have to rewrite the record books. So the first bacterial genome sequence wasn't sequenced by that American arrivist Craig Venter. It was sequenced by Fred Sanger. Good old British Nobel laureate Fred Sanger sequenced the first bacterial genome in 1981. When we look at the most successful bacterial clade on the planet, Jack Gilbert's not here, but let's just make it clear it's not SAR11, it's the Mitochondriaceae. And back of envelope calculations suggest we've got 10 to the 26 representatives just in us humans, and then you've got millions of other animals and plants as well. So there must be 10 to the 30 something of mitochondria on the planet. Henrietta Lacks, this African-American woman who was the source of HeLa cells, is probably the most abundant mitochondrial human lineage on the planet, probably 10 to 17 HeLa mitochondria in existence currently. Another issue is, of course, that if we say, what's the smallest bacterial genome, we've got a serious philosophical problem now. Because, okay, we can say, well, the Plasmodium mitochondrial genome is only 6 kb, very small. There's another one, this early branching dinoflagellate, that actually has fewer coding sequences in its genome, but it is still a piece of DNA. But we have a reductio ad absurdum here because some mitochondrial derivatives have actually thrown away their genomes, completely outsourced the whole lot. The mitosomes of Cryptosporidium, hydrogensomes of Trichomonas. So does it actually make sense to say that the smallest bacterial genome is 0 base pairs? Or does even the most fervent cladist have to accept that something's funny here? Let me just quickly finish. Obviously, we need to rewrite the textbooks. Let's give bacterial nomenclature to all the genes and proteins in mitochondria. Two applications that I think are quite cogent. So, okay, we've got all these bacteria living inside our cells. How come our innate immune system doesn't recognize them as bacteria? We know that it recognizes these so-called pathogen-associated molecular patterns, or PAMPs. Well, one answer is that actually the mitochondria have thrown away a lot of the PAMPs. So they don't have LPS, flagellins, peptidoglycan. But some of these PAMPs do persist in mitochondria. So we have mitochondrial proteins, like those of non-organella bacteria. Notice I'm adopting the correct nomenclature now. Are N-formulated. And these formyl peptides are known to activate the innate immune system's formyl peptide receptors. And mitochondrial DNA, like other bacterial DNA, is rich in these unmethylated CpG dinucleotides, making it a ligand for TLR9. And so, actually now, when you configure it this way, you have an explanation as to why the response to trauma actually looks like the response to sepsis, because it's pretty much the same thing. Because the innate immune system does recognize mitochondria as bacteria. And this guy here, Karl Hauser and his group, got a Nature paper on this. I just checked, it's been cited nearly 300 times in the last 2 years since it appeared, pointing out that this is the case. And he actually coined the term DAMPs for these things released from the mitochondria that are in fact mitochondrial PAMPs. And so this provides us a nice satisfying explanation as to that should have been obvious earlier. If we'd have really taken seriously the idea that mitochondria are bacteria, maybe we'd have got to this stage earlier. Another ramification of this is if we treat mitochondria like bacteria, we might actually come up with ways of manipulating them. And what if we turn these in-house intracellular bacteria into cloning hosts as versatile as E. coli? What new opportunities would arise? Well, actually, at the moment, there is no practical way to re-engineer vertebrate mitochondrial genomes. But this guy here, Mike Koob, has actually shown that you can use bacterial conjugation to get DNA into mitochondria. The bacterial conjugation system has evolved to actually get DNA through those two membranes, same as what you see in mitochondria. And in fact, they've produced, he and his associate Yoon have actually produced a string of papers showing that this approach works, and they're gearing up to actually start re-engineering human mitochondrial genomes. And they've even just had a paper out showing that you can take invasive bacteria that have, say, the Shigella invasion apparatus and get them to conjugate mitochondrial sequences into mitochondria inside cells. So if you want to look for the future, someone's got a mitochondrial disease, you inject an engineered bacterium which invades their bloodstream, invades their bone marrow, and reprograms all their mitochondrial genomes to be what they should be. It doesn't have to stop there. We can put whatever we like into that genome and create transhumanist, next generation humans, get rid of the requirement for vitamin C, put that metabolic pathway in, whatever you like. So, maybe you're not ready for this phylogenetic fundamentalism, but I think I've laid out the arguments as well as I can. I think that there are interesting ramifications when we start thinking this way. This collaboration's been going on for 2 billion years in the natural world. I think the real challenge for synthetic biologists is to stop mucking around making booleans, gates and all that kind of stuff, re-engineer our inner bacteria and make them useful for us. That's me finished. Thank you.
So is everyone in agreement? Question here? Wait for the mic, please. Sorry.
If you actually force a conjugation between mitochondria and bacteria, can you have the recombination of such DNA into the mitochondrial genome, and then because are the systems of recombination still available in this organelle?
I don't think so, but you don't need to do that. These genomes are so small that you can just, a la Craig Venter, you could just make a 16 kb or 20 kb molecule and conjugate that in. I think would be the goal. As I say, I've not actually done any of this. We've been trying to import DNA in my group, but only through a project student. I've got any publication track record on this, but also among plants, I think it's much more tractable to do things to mitochondria. It's just among animals, and particularly vertebrates, that seems to be an issue in getting stuff in.
Because I was curious about it some time ago, and now I don't know if someone succeeded in actually expressing something external in mitochondria.
Well, Youb and Kuna have actually put in DNA through conjugation and shown that it's transcribed. They have also shown that antibiotic resistance genes work in mitochondria, preparing the way for actually having genetic constructs that they can select and so forth. But it's still at very early days, and we don't know. Maybe that there's all sorts of hidden things out there, epigenetic modification of the genome that we ignore or whatever, that means that actually it won't work straightforwardly. But I think it's a provocative idea to try, and I just find it strange that there's not more effort going into this, actually. It seems to be these two guys in the US seem to be the pioneers, the only two doing it.
Crystal?
I think an interesting, possibly slightly different way of posing the question is in terms of sort of evolutionary fate or evolutionary trajectories or dynamics. Do you think it's possible to disentangle things which determine evolutionary dynamics, like fitness and so on, to disentangle the fitness of the organelle from the fitness of the cell or the organism?
I mean, that's an interesting question. If you look at it from the point of view of the organelle, apoptosis, we always think apoptosis is something that eukaryotic cells do to keep the multicellular organism going. You could argue that apoptosis is just an addiction mechanism. You know, if the mitochondrion gets damaged, it says, right, okay, if you let me get damaged, I'm going to kill you. And it's a way of making sure that the eukaryotic cell can't survive without the mitochondrion. There's some interesting work being done with this canine. There's a transmissible cancer in dogs, and that cancer is able to pick up mitochondria during its travels through wolves and dogs and foxes, and you end up with this mishmash of different mitochondria in there. And the argument is that the cancer itself, its mitochondrial lineages lose fitness, and they start to degenerate, and it has to pick up new ones. That may be a just so story. That's the argument that's been put forward in a recent paper. So yeah, there's a lot of interest in genomic diversity in mitochondria in humans, in cancers and things like that. That's quite a hot topic, I understand, at the moment. So yeah, it's interesting.
We'll just take one more question.
That was a brilliant talk. I really enjoyed it. Thank you so much. So, you know, your last comment is that biologists stop making Boolean gates and stuff.
I was being provocative there.